Omega-6 fatty acids, particularly linoleic acid, have long been considered essential components of a balanced diet, often found in vegetable oils like soybean and safflower oil, as well as animal products such as pork and eggs. However, a recent preclinical study has raised serious concerns about the potentially dangerous effects of this seemingly innocuous fat, particularly in relation to one of the most aggressive and challenging forms of breast cancer, triple-negative breast cancer, TNBC. The findings of the study, published in Science on March 14th, revealed that linoleic acid can activate a major growth pathway in tumor cells by binding to a protein known as FABP5. This discovery is concerning, especially as it highlights the specific risk to patients with TNBC, which is notoriously difficult to treat due to its lack of estrogen, progesterone, and HER2 receptors. Unlike other subtypes of breast cancer, TNBC lacks targeted treatments that focus on these receptors, making it significantly harder to manage and treat. In the study, researchers found that linoleic acid activates the mTORC1 pathway in TNBC tumor cells. This pathway, known for regulating cell metabolism and promoting cancer cell growth, is particularly activated by the presence of FABP5, a protein that is abundant in TNBC but largely absent in other breast cancer subtypes. Through experimental studies with animal models, the researchers demonstrated that mice fed a high linoleic acid diet saw increased levels of FABP5 and TORC1 activation and enhanced tumor growth. These findings suggest that high omega-6 intake could promote the aggressive growth of triple-negative tumors, exacerbating the challenges of treating this difficult cancer type. Furthermore, this study is the first to establish a clear biological mechanism that links omega-6 fatty acids, specifically linoleic acid, to cancer growth. Although previous studies have suggested a potential link between omega-6 and various diseases, the precise biological pathways involved had remained unclear until now. By showing that linoleic acid enhances cancer cell growth via the FABP5 and TORC1 pathway, this research provides much-needed clarity on the role of dietary fats in cancer progression. The implications of these findings are both alarming and complex. With the prevalence of omega-6 fatty acids increasing in Western diets due to the widespread use of seed oils in processed foods and cooking, the potential for these dietary fats to contribute to the rising incidence of cancers like TNBC cannot be ignored. Since omega-6 fatty acids are essential for many bodily functions, the key issue appears to be not their presence in the diet, but their excessive consumption, which has significantly increased over the past few decades. One major concern is the absence of effective targeted therapies for TNBC. Currently, treatments for this aggressive subtype are limited and often involve chemotherapy, which can be highly toxic and less effective than therapies that target specific receptors in other breast cancer subtypes. The identification of FABP5 as a potential biomarker for TNBC opens the door to more personalized treatment options. However, this also raises the question of whether dietary modifications, such as reducing linoleic acid intake, could play a role in the management of the disease. While the idea of using dietary adjustments as a therapeutic strategy may sound promising, it also raises concerns about oversimplifying the complexities of cancer treatment. Altering one's diet may not be a sufficient strategy to counteract the effects of aggressive cancer growth, and patients may be misled into thinking that simple lifestyle changes can replace the need for more advanced treatments. Moreover, the study's findings also suggest that the FABP5 and TORC1 signaling pathway might be involved in other diseases, such as prostate cancer, obesity, and diabetes. This raises additional red flags, as these conditions are already linked to modern lifestyle factors, including poor dietary choices. The idea that a common dietary fat might contribute to the growth of multiple types of cancer and chronic diseases calls for urgent public health awareness. The continued overconsumption of omega-6 fatty acids, especially from processed and fried foods, could be a hidden factor fueling the rise of these diseases, which are already at epidemic levels in many parts of the world. However, while these findings are deeply concerning, they also underscore the importance of more comprehensive research into the impact of omega-6 fatty acids on human health. The study does not establish a direct cause and effect relationship between omega Omega-6 intake and cancer progression in humans. It is crucial that further clinical studies are conducted to validate these findings and explore potential strategies to mitigate the risks posed by excessive omega-6 consumption. Additionally, it is important to recognize the limitations of focusing too heavily on a single dietary component in cancer prevention. While the study highlights the potential dangers of excessive omega-6 intake, it does not address the broader context of diet, lifestyle, and genetic factors that influence cancer risk. Relying solely on dietary changes may divert attention from other crucial aspects of cancer prevention, such as regular physical activity, maintaining a healthy weight, and minimizing exposure to environmental carcinogens. In conclusion, the discovery that linoleic acid can promote the growth of triple-negative breast cancer is a critical step in understanding the relationship between diet and cancer. While it offers valuable insights, it also raises several warnings. The increasing prevalence of omega-6 fatty acids in the modern diet, coupled with the potential risks to health, demands a more nuanced approach to dietary recommendations. As we seek to understand the complexities of cancer and other chronic diseases, we must avoid simplistic solutions and focus on a holistic approach that incorporates multiple factors, including genetics, lifestyle, and environmental influences. The findings of this study should serve as a wake-up call to both researchers and the public about the potential dangers of excessive omega-6 consumption, particularly in a world where processed foods dominate the diet.